Hi, everyone. Welcome to Foresight's Biotech and Health Extension Group. I'm really excited today to have Logan with us. Logan is usually a guest in our newer tech group and for really good reasons that we might get into in the Q&A if we have time. But I'm really excited to welcome you today to present on viruses inside walls, which is a very sci-fi topic. I love it. When this is a sci-fi title, but you will put a little bit more meat to the bone. Please take it away. I'm going to share a lot more info about you in the chat, and I'm going to be in the chat for questions, which we'll hopefully get to. So again, as usual, we are prioritizing questions from the chat for the Q&A later, but you can also just raise your hand. Please take it away. Thanks a lot for joining.
All right, everyone. It's great to be part of this virtual seminar with Foresight. And I'm going to be talking today about a different topic than I have to talk about with Foresight. I'm going to be talking about my gene therapy research. Specifically, I'm going to be talking about a new way that I've developed to avoid pre-existing immunity to a viral vector called AAV, or adeno-associated virus. And the sort of relevance of this to the longevity group is that there's AAV, as I'm sure you're aware, or at least some of you may be aware, is a leading gene therapy delivery system that is used in a lot of the most cutting edge gene therapies today, and will likely be used soon in some sort of broader. Currently, it's used in a lot of sort of single gene diseases, but at some point in the near future, it may be used in broader contexts where it could treat things like aging and the various associated factors that go into that. With that, I'll mention that 30 to 60% of patients already have pre-existing immunity. They have antibodies in their bloodstream and throughout their body against adeno-associated viruses. And this can be very problematic for gene therapies because it basically means they're not really able to benefit from the treatments that the gene therapy can offer. Additionally, and this is especially important for aging, is that AAVs can't be re-administered since they essentially act as a vaccine against themselves, and neutralizing antibodies will develop against the first dose after the first dose. And therefore, you can't really administer a second dose since there's already going to be a immune response a strong secondary immune response that will be mounted against them. Especially if you want to do repeated therapies or therapies to target more than one gene or something like that, over time, it would be very difficult to use traditional AAV gene therapy. And people have talked about things like lipid nanoparticles, of course, but lipid nanoparticles have their own issues in that they're not very, very targetable, and they have a pretty low efficiency, generally speaking, for transduction of cells. And this can be especially problematic in the case of gene editing therapies, which I think would be likely to be highly relevant for longevity. There are also some increasing concerns about the immunotoxicity of high-dose AAV gene therapies. And this is something that has actually resulted in a few deaths where people have had very high doses of AAVs injected in some clinical trials or some trying to treat some very unfortunate diseases that they're suffering from, and there have been a few deaths. And so there really is a need to find a way to deliver these AAVs without having these problems. So people have tried, and there's been some good progress made. So one way that people have tried to engineer AAVs to circumvent the neutralizing antibody problem, as it's called, is through directed evolution and/or machine learning that is able to essentially modify the capsid of the virus to essentially be different from what the pre-existing immunity is able to target, making a variety of mutations that lead to different structural features and so on and so forth the antibodies can't bind. However, this method, although promising, is limited due to the remaining cross-reactivity of antibodies, and it still doesn't allow re-administration unless you create sort of a series of different capsid variants, which can be very difficult to get that many different variants approved from a regulatory perspective. In addition, some of these approaches can disrupt or at the very least limit AAV transduction into cells, that's uptake into cells, and production. So it can actually make it more expensive and more difficult to produce the correct amount of AAV or enough AAV to use for patients. Another method that people have tried is called plasmapheresis, which is typically coupled with immunosuppressive drugs. It actually involves taking out a large portion of the patient's plasma. This is invasive, expensive, and risky, as you might imagine, actually removing a huge amount of the plasma from a patient. And then there's exo-AAVs, which are exosomal associated AAVs. So these are promising, but there are some issues with poor yields, purity issues, and then also inconsistent titration or counting methods to actually know how many of them are in each dose. And these have been very problematic in that they've prevented clinical translation. And this was invented in 2012, so it's been quite some time since they were first developed, and there hasn't been a whole lot of progress. To move on to the solution that I'm developing, protein bolts. So what are protein bolts? First, I'll give a little bit of background on this. Bolts are these large 70 by 35 nanometer protein nanoparticles that are found in most eukaryotic cells. Actually, quite a lot of them are made in each eukaryotic cell, typically. And as a result, they're essentially immunologically invisible in mammals since they're viewed as a cell antigen. So they're not really an antigen at all in that sense. And yeah, there's actually, I believe, 10 to the fourth or 10,000 plus copies produced in each of our own cells, sometimes even more. And bolts also have a few useful properties. So they show a strong natural tropism for targeting phagocytic cells like macrophages and dendritic cells, as well as activated T cells, which aren't technically phagocytes, but they also show tropism for those. And then, however, they're not limited to this. So bolts are also endocytosed by other cell types, albeit at a lower efficiency, though as you'll see, it's still pretty high, generally speaking. They seem to be taken up into a lot of different cell types very well. But all that said, bolts can actually be modified at the C-terminus, which actually ends up being the sort of those top and bottom portions, or in this case, the left and right, this sort of circular portion up here, and then down at the other end. Those are the C-termini with peptides or antibodies. And these peptides or antibodies can retarget the bolts for uptake into other cell types. And this is stuff that's been done before for delivery of proteins and small molecules using bolts. And so how do people put bolts, things inside of bolts? As you can imagine, to use these for delivery, you need to package something into them, whatever you want to deliver. And so typically what happens is that one will make a fusion with a peptide called INT, which is part of a protein that's found in endogenous bolts, a PARP protein, a VPARP protein, actually, that is polyadenyl ribosylase. And so that protein is typically found in bolts. We don't know what its actual function is in bolts, but nicely enough, it sticks to the inside of bolts around the waist region. And so you can actually use this 166 amino acid peptide to bind to the waist region of the bolt. And nicely enough, the bolts will actually breathe slightly, so they expand and contract. And these cargo INT fusions will just spontaneously slip inside of the bolts. Another factor that should be noted is that it can be useful, though not strictly necessary, to use endosomolytic proteins to release cargo from bolts into the cytosol after they've been taken up into cells. And if you, there was actually a paper published by my collaborator, Leonard Rome, where they used a adenovirus protein called protein 6, which was packaged into bolts and then was used to enhance the escape of the bolts from the endosome after cellular uptake. And this will come in a little bit later in that they actually have their own endosome escape capabilities. And as you'll see, this can be helpful. However, it should be noted that even without endosomolytic proteins, some bolts can get out of the endosome, but it's not as efficient. Just to give a little more background, and I promise I'll get to the good parts soon, bolts have shown some good efficacy for shielding small proteins from immune responses and allowing them to be delivered into cells. Interestingly, they've actually been used to deliver antigens that are used as a vaccine, such as the chlamydia major outer membrane protein. And this has been used as an intranasal vaccine, and this has been tested in mice. They've also been used to deliver chemokines like CCL21 into tumors, and this triggers the infiltration of the tumors by immune cells. And then finally, there have been just some other, perhaps somewhat earlier stage papers that have been published on using volts as shielding and delivery agents. However, volts have not so far been able to package nucleic acids except for a few small interfering RNAs. Interestingly, larger nucleic acids typically will get spat back out of the volt they're packaged. Leonard Rome, I mentioned him before. He's the sort of the pioneer of volt technology. He's actually an emeritus professor, and he has shown that his group has attempted in the past to package larger nucleic acids like messenger RNAs or DNAs containing genes into bolts, and they actually will go in and then get spat back out. And they don't really know why this is. It doesn't seem like it's an electrostatic thing or anything like that, but that's what has happened in the past. And however, I'll mention in a moment that sort of I've been able to overcome that issue. So what if we could use volts to shield AAV for neutralizing antibodies? And I've drawn this nice schematic here to just visualize the process where volt AAV complexes would be amongst the glue of antibodies, and then however, the volts would shield the AAVs from the antibodies, be taken up into cells, disassemble at the low pH of the endosome, which is something that disassembles volts. Volts disassemble at low pH. The endosome acidifies, and so this will release the AAVs, and then the AAVs will then go through their usual pathway of endosome escape and trafficking to the nucleus for gene expression. AAVs actually encode a protein that has a phospholipase A2 domain that lyses some of the lipids in the endosome and facilitates endosomal escape. Yes, how am I doing this? I need to get the AAVs inside the volts. And you heard me discuss the INT peptide previous. So I'm actually using something called a molecular glue or SpyTag SpyCatcher technology. There's also dog catcher technology, which is basically the same thing, but an orthogonal pair of peptides. What SpyTag and SpyCatcher do is they react with each other in a highly efficient and highly specific fashion to form a covalent isopeptide bond, which branches off of the main chain of the proteins. And so when you mix them, they react and link together. And since they're proteins, you can fuse these or insert these into other proteins. And so what I've done is I have inserted the SpyTag, which is a 16 amino acid small peptide, into the VP2 protein of AAV. And then this can be displayed on the surface of the capsid in about 5 to 10 copies or about 5 copies actually, give or take, because it's a stochastic number, but it's a distribution, that is. But about 5 copies on average of the SpyTag will be displayed on the AAV capsid. And then one can use SpyCatcher. So I used SpyCatcher fused to the INT peptide and showed that it was able to bind and react with the SpyTag on the AAV, thus linking INT onto the AAV surface. And over here is just a gel showing that the linkage has happened based on the molecular weight shift of the bands. The SpyCatcher 3 INT fusion protein, which is down at the bottom here, this is another gel that just shows that it has the correct molecular weight. And so from there, I'll talk about some of the exciting stuff. AAV, when linked to INT, it actually spontaneously packages into the volts, which is really cool because one wouldn't really expect it to actually, with something this large, to be able to slip inside of volts. And yet it seems that it does indeed happen. So what I've done is I found that actually just room temperature incubation overnight at a 1:5 AAV INT to volt ratio induces uptake of or packaging of the AAVs inside of the volts. Not 100% efficient. So there are, at least with my initial experiments, it seems like only about 10% of the AAVs get into the volts. But I've since then used a larger amount of SpyCatcher INT. So instead of a 2-fold excess, I've used a 20-fold excess, and that seems to increase the packaging to about 50% of the AAVs getting into the volts. But that is something that's still ongoing. So yeah, I think it's likely that it'll be possible to increase it to nearly 100% at some point in the relatively near future. This is the first demonstration of packaging an entire virus into a vault. It's also the largest cargo ever packaged in vaults. And it's the first method that succeeds in packaging nucleic acids, that is, other than the small interfering RNAs, that succeeds in packaging these larger nucleic acids into vaults. And in this sense, it is actually that the AAVs contain these 4.7 kilobase genomes that can be used to encode things, and they are packaged into the vaults with the whole AAV complex. So here's something really exciting. So this is electron microscopy images, transmission electron microscopy images of a AAV INT over here on the left, being that have packaged into some of the bulbs here. So you can see the AAVs as those little almost hexagonal particles there that are packaged into the vaults, which look like little jars. And then on the right is control, where I had the AAVs linked to spine catcher, but no INT peptide, and they don't package into the vaults in the control. And yeah, there you go. So that was the first big thing that drove this project forward. The next was, before I move on to that, here's just some more cool pictures of them that are a little closer up. So you can see some of these representative Volt AAV complexes. One really interesting thing is that occasionally, maybe 1 in 100 times, Volt AAVs will actually include 2 AAVs inside of a single Volt. So that is something that might be able to be exploited in the future if the frequency of that could be increased. That might be able to be exploited in the future to deliver larger cargoes or more genes or so on and so forth, which again could be very useful for aging if one was able to deliver more complex cargoes. This is just a wider field view of the Volt AAV packaging, experimental and control groups. I only include this because in the experiment that I'm going to show you, it explains why the transduction efficiency is lower, and I'll in some of the cases, and I'll mention why that is. It just corresponds to the approximate percentage of AAVs that are packaged into vaults, but that'll become clearer momentarily. Another thing that I want to show you before I move on to the cellular transduction experiments is that AAV INT spontaneously packaged into vaults can be reconstructed using cryo-electron tomography. And this is really exciting because it shows us an actual 3D representation of what this looks like. You can see this cutaway view. It's actually, the bolt is intact, but it's a cutaway view since there's something called the wedge reconstruction problem that I'm told it happens with small particle numbers in cryo-ET. And so we can't see the entire every angle, but we can see this cross-sectional view. And that's helpful anyway, since it allows us to look in and see the AAV. So this is what they actually look like, at least roughly speaking. And that's really cool. All right, now I'm going to get to another cool thing, which is that Volt AAVs are indeed able to evade neutralizing antibodies, at least in vitro so far. So Volt AAV complexes will indeed transduce cells in the presence of a neutralizing serum that has actually been taken out of mice and put into the 96 well plate that I was using. So this serum was from mice that were immunized with AAVs that have the spy tag. And then the serum was taken and I mixed it with my AAVs and then also with my Volt AAV complexes. So to just explain this a little bit more, Volt AAV, or VAAV in this case, is what I'm labeling it as. This group is the Volt AAV complexes. This group over here, and by the way, people should feel free to ask questions at any point along the way if you'd like. Yeah, so this group is essentially the Volt plus AAV where there's not packaging because the INT peptide is missing. So this corresponds to this group here where it's Volt plus AAV but no packaging. And yeah, and then this is AAV alone, and this is just phosphate buffered saline. And then this row is with anti-AAV serum, and this row is without anti-AAV serum. And what you'll see is that, yeah, here the Volt AAV complexes are able to evade the serum in order to transduce cells. Volt plus AAV actually seems to have a slight effect of being able to get past the antibodies, but not nearly as strong as the Volt AAV packaged complexes. And then AAV alone, there's not really any transduction at all. Interestingly, it seems like the bolts actually enhance cellular transduction quite a lot as well. In addition to having the effect of circumventing neutralizing antibodies, they also seem to enhance transduction even without antibodies. So you can see down in this row with this row with the without serum, without neutralizing serum, there's really a lot of transduction in the Volt AAV and Volt plus AAV groups, interestingly, both of them. So just the fact of mixing the volts with the AAV seems to really enhance transduction. Whereas AAV alone, that's the main control, you can see there is transduction, of course, here is about 20%, but it's not as high as in the Volt AAV groups. This is the same thing. I just quantified this same experiment using flow cytometry. And here you can really see the differences. So here it's about 4% transduction in the presence of anti-AAV serum with the Volt AAV complexes. And that goes back to that wide field view that I showed you. So I was saying about 10% of the AAVs were actually packaged into volts. This kind of explains why it's only about 4% that are transducing here, because there's probably the rest of the AAVs that aren't packaged into volts are getting wrapped up by the antibodies or absorbed to the antibodies and being unable to transduce the cells. And really, this is what we're looking at here is the ones that are actually inside the bolts are able to circumvent the neutralizing antibodies. However, there does seem to still be, even with AAVs that are mixed with bolts but not packaged, perhaps due to the transduction enhancement effect, about 1% of cells were transduced with the Volt plus AAV group. And then we don't really have hardly any transduction in the AAV only, of course, because the antibodies are getting rid of those. And then interestingly, without serum, though, we do see higher transduction levels. Again, because the serum, the packaging here was inefficient. I think that if we had 100% efficient packaging, we would see something closer to what we see over here. So anyway, that's just to clarify that. But you can see really it is quite impressive. About 85% of the cells are transduced with Volt AAV and Volt plus AAV, but only 20% with the AAV only control. So this is really interesting. Are people in the chat asking questions right now? I can't see it, but
we have one, but we can also wait until the Q&A, depending on what you prefer.
Yeah. Yeah. Okay. I see what the question is. Yeah. So when I spoke to Leonard Rome, he said that he has actually seen this kind of transduction enhancement effect in the case of just naked plasmids. When transfecting cells with plasmids, it seems that something similar happens. So the plasmids aren't actually inside the bolts, but they're mixed with the bolts, and then they seem to get into cells more efficiently. And this is hypothesized to be something called the bystander effect, where there's actually sort of absorption to the outside of the bolts, and then the bolts are efficiently eaten up by the cells, and this gets whatever has stuck to their outsides into the cells more efficiently. And that's not like completely verified, but that's one of the main kind of hypotheses as to why this may happen that's out there. But really, it's just cool that it happens in this case too, because this could be very useful for gene therapy. In particular, if this transduction enhancement effect persists into in vivo tests, there may be a way of leveraging that to use lower doses of AAVs, which could both mitigate immunotoxicity further and lower the cost of AAV gene therapies, because you would need to produce less per treatment since you'd be using a lower dose. And this could be really important, especially for using AAVs on a large number of patients, because right now AAV gene therapy is very expensive, in part due to manufacturing issues and in part due to insurance issues because of the small number of patients it's being used on. But really, there is some question as to how we can scale up AAV manufacturing to the point where we could actually be giving large numbers of people AAV treatments. And this kind of transductional enhancement could help address that problem, at least partially, by allowing us to use less AAVs overall and thus needing to produce less AAVs. I'm going to, there we go. So that's about as far as I've gotten so far. However, I am in the process of starting to put together an in vivo study with mice. Specifically, I want to go for the lowest hanging fruit first, just to demonstrate that it can work. I want to show that both AAVs will be able to transduce the liver in mice that have been pre-immunized against AAVs. And this will be a similar test to what I showed in vitro, but in this case, we're going to be looking at the liver. Because the liver is the most efficiently transduced organ. I see there's another question in the chat. To clarify, do the bolts also serve to prevent the immune system from recognizing the AAVs initially, preventing the immune system from acquiring that immunity? Yeah, that's a good point. I think that it would likely be that it would mitigate antibody responses to a large degree. However, and even the development of further antibody responses. However, I would think that there still would be some buildup of immune response after repeated administrations, because a few AAVs might still be kind of out there and triggering immunity, especially if you don't have 100% packaging. And so that could potentially be something that would happen. However, the bolts should shield from antibodies, generally speaking, even if there are antibodies out there. But I should also note that there's also the cellular immune response, the immune response that involves sort of intracellular stuff, where the pieces of the AAVs would be chopped up and presented on MHCs and so on and so forth. That could still be somewhat of an issue, but generally speaking, it's recognized in the field that if we could completely get rid of the antibody response, it would go a really long way to mitigating immunity, generally speaking, even if there is still a cellular immune response. Yeah, and Micah is saying that, yeah,
the two mice are different.
Oh, yeah, that I just that's the only difference.
Yeah, no, I
Well,
just
like,
I just wanted them to look like they're
different. That's
Do I-
nice. Sounds like a nice attention to
I
detail.
probably usually am.
yeah. I just drew them that way because I want them to not just look like two copies of the same mouse. Yeah. So anyway, Micah was looking at was talking about the free AAVs potentially triggering the formation of neutralizing antibodies, and yes, as I mentioned, that probably would be the case, especially if it wasn't even a few free AAVs were out there that had packaged. But the idea is that hopefully then, even if you wanted to do repeat administration, the bolts would still shield the AAVs that are packaged from the antibodies even after they develop further. And yes, back to the in vivo study. I see there are some more questions in the chat, but I'll check that out.
Yeah, I think maybe we take the questions at the end because they're
Sure,
just going to keep on coming.
Yeah, that's true. That's true. Yeah, I just wanted to make sure that people would get things clarified if needed. But yeah, I'll probably address them at the end at this point. Yes, working on this in vivo study, I actually did do a previous attempt at an in vivo study that was aimed at a vaccine application, which I think was a mistake because I ended up using intramuscular administration, and I did not check the liver gene expression. And I think that and so that whole experiment actually ended up being null in a positive control. But it taught me a lot about the process of running an in vivo experiment. And so I think that I actually have some degree of optimism, at least, for the likelihood that this next in vivo experiment, which is aimed at lower hanging fruit, just this liver transduction thing, will yield some interesting results because, for a variety of reasons. Yeah, and then just the second bullet point here is that I'll also observe to see if there's a transduction enhancement effect in the both late AAV and full plus AAV groups. And I'm also switching to a different AAV serotype called AAV DJ. And I'm going to be using a different tag called dog tag, which is better for being inserted into the middle of peptides. I've actually used this AAV DJ dog tag for a different project that I'm working on, and it has shown better reactivity of its dog tag with the dog catcher. And so I think that there's promise that this AAV DJ might actually package even better than the AAV9 serotype I was using previously. And so I'm trying to get some AAVs produced for this using a core facility, trying to get enough for 10 to the 11th per mouse particles. I'm currently forming a spin-off company that is called Cathedral Therapeutics Incorporated. I do have a full patent that has been, it's being processed by the USTPO, but it was filed on July 17th on Volt AAV technology. It should be public in about 5 months, maybe about 4 months by now. And I'm actively seeking some pre-seed funding to support early stage development. I have gotten a little bit from a group called Soma Capital, which it's going to be transferred soon anyway, which is very good for some of the early stage stuff that I'm working on. But yeah, I'm now working on spinning off this company. I'm trying to get large, and hopefully once I have it, the animal proof of principle, I'll be able to support larger fundraising routes that can facilitate moving into preclinical efficacy and toxicology studies and then eventually clinical trials. And with that, here are my acknowledgments, and I will answer the many questions in the chat that I think have popped up.
Fantastic. Then let's hit the road. We had a question. Jose, you asked one, I think, a while ago. I'm not sure if it went answered. I know that one of your smaller questions went, but the early one, if you want to repeat it, feel free to unmute yourself.
I know. I think he has to answer it. It was like yesterday. It's like his hitchhiker effect. So it's well answered. Really good talk. Thank you.
Okay, awesome. Yeah, I agree. Very good talk. Totally forgot to say that. I was just trying to get through all the questions. But yeah, it was a really good talk, Luke.
Sure, thank you. Yeah, and by the way, just as an add-on to Jose's question, so it probably, in this case, it probably will have some off-target organs, but liver transduction generally, especially for AAV DJ, liver transduction is by far the strongest transduction that would happen. And so I'm going to be focusing on analyzing the liver because that's going to yield the most visible results at this point. However, it could be potentially valuable to look at some other organs such as the spleen, the lungs, etc. But there may also be some limitations on sort of the scale of the study that may make that a little more difficult at this point in time. But at some point, eventually, I'm going to do full those preclinical toxicology and efficacy studies. I would want to do full analyses of how different serotypes biodistribute to different organs. The next question, I think, Allison, was...
We have one from Martin from a long time ago. If you want to unmute Martin, if you can, feel free to. Otherwise, I'm happy to read out your question.
Sure.
Oh,
What was that?
in your diagram, it looked like the protein, but I've seen some other pictures where it looks like more like beta structure. Could you comment on that?
Yeah, so the volt, I can actually go back to that slide. So there are beta repeats in volts, and there's also alpha helices. It's the sort of that top region where the alpha helices are present. You can see that they wind together. This is actually, there's 37 copies, I believe, of major volt protein that sort of lace together here. And then each one has an alpha helix that is up here at the top. And then down towards more the waist domain, there's these beta repeats that are beta structures. Yeah, hopefully that answers your question.
Are the helices amphipathic?
Amphipathic? I don't know, actually. I think in terms of I'd have to look at the charge distribution of the volt.
Okay.
Is there a specific reason?
Thank you. I was just thinking about the self-assembly.
Sure. Yeah, that would make sense.
Cool. Next one up with Micah.
There are you can download the volt structure and look at the charge and hydrophobicity and so on and so forth using things like Primal or whatever. If you're curious. Yeah. So what was the next question?
Micah raised his hand.
I have a quick terminology question followed by a real question. Terminology question: would bolts be considered extracellular vesicles? They fall into the same category.
No. So extracellular vesicles are typically composed of lipids. They have proteins in them, actually lots of proteins, as transmembrane proteins and probably proteins on the inside as well. But these are very different from extracellular vesicles.
Okay. And the actual question is this limited to AAV, or could this also work with like other viruses like CMV? And can it work with things like various types of plasmids like minicircles or dumbbells? Or is there something specific about AAV that makes this reasonable in that case, but not in the others?
Right. The reason that AAV is a prime candidate is, for one thing, AAV is it has such a strong clinical profile and has seen so much use already and has had a lot of successes. Also, AAV is small enough that it's smaller than the volt, so it actually can fit inside the volt. If you try to use cytomegalovirus or adenovirus, those are both actually larger in dimensions than the volt by quite a bit. So it wouldn't really make sense to try to put them inside since they're bigger. You could imagine potentially decorating the outside of a larger virus with multiple copies of a volt, but actually packaging it inside doesn't really seem very likely to happen since it's that size. I think, what was the other part of the question? Sorry.
could things like other types of plasmids, mini circles or dumbbells or
Plasmids.
something like that also work potentially?
Yes, there is some renewed interest in that. As I mentioned earlier, the past attempts at putting plasmids inside of bolts has led to the DNA being spat back out. Same thing with messenger RNAs. And so it doesn't really stay inside the bolts. And again, they don't know why, but there are some renewed efforts at trying to package DNA alone into bolts without a virus. And so that could be another good approach. However, having the virus does add benefits in that it has all the virus's machinery for intracellular trafficking, for endosomal escape, and so on and so forth. I think, yeah, you can see here. So the virus is able to facilitate endosomal escape and facilitate nuclear trafficking, which is something that sort of has an added benefit relative to just a plasmid on its own.
Awesome. Are there any other questions, comments from anyone? Feel free to raise your guys' hand.
I think
we also have
Michael
another
asked
question
a
in the
question.
chat from Michael. Yeah, Michael isn't here anymore, but I can read it out.
Okay.
What are your thoughts on IDAS protease, mycoplasma protein M to prevent overcome immunity?
Trying to think if I remember what those do. I actually don't think I'm familiar with those, so I don't think I can really address that question.
Cool. Okay, we won't make you speculate. That's totally fine.
Yeah. I'm trying to think if I remember, but yeah. Anyway,
That's okay.
Jason?
Next question is Jason.
Yes, thank you. Great info and presentation. I was wondering, in terms of targeting the liver, when you target it with the AAV and the vault, how do you target that liver without having off-organ effects? And are you targeting all cell types in the liver, or only like a subset of the cell types within the liver?
Sure. So I think I answered a question similar to this a moment ago, but I can say again, yeah, the AAVs and volts, actually, both AAVs and volts will, because the liver is the organ that takes in things that aren't usually there, anything really, even other things like other proteins and drugs tend to go to the liver more often than they go elsewhere. Sure, there's also other places that if you inject something in the blood, things will go elsewhere as well, but the highest concentration is usually going to be in the liver. And so this can be great if you want to treat something that has to do with liver disease. If you want to treat something else, you're going to get some transduction of the liver regardless. But you can target things so that they also go elsewhere to some degree of strength. But yeah, the liver transduction thing is really just a vehicle at this point to show that it works. And it's the lowest hanging fruit because the transduction is going to be strongest in the liver. And that's just really a property of our physiology. Does that answer your question?
I think so. Just a quick follow-up. If say the AAV and the vault get to the liver, like in your picture here, it looks like it just goes right through the cell membrane. Is that something that happens in all the cells within the liver, or is it not known if
right.
some of them maybe just bounces or? Yeah.
So this is showing endocytosis, where the cell sort of gobbles things up into an endosome. But yeah, there may be some cell types in the liver, especially hepatocytes, that are more likely to take up the bolts than other things. Like I think there's certain dendritic cells that are liver specific that are probably actually, because they're volts and the volts like phagocytes, they might go into those as well. But there are a variety of cell types in the liver, and some of them may take up both AAV complexes more strongly than others. So that is a good point. But it doesn't really matter in the context of my current experiments. But like if I was looking to treat a specific disease, or address in this case, a certain genetic factor that may contribute to longevity, then looking more in detail at what cell types I want to target and what cell types I want to avoid would become more of an important factor. And obviously at some point I'm going to be doing that, looking into a specific disease target down the road. I've been considering alpha-1 antitrypsin deficiency, but as a sort of early target. But yeah, for now, it's the targeting is a second priority. But yeah, targeting is important, generally speaking. It's just not quite yet for the proof of principle.
Okay, thanks. Appreciate it.
Yeah. And someone asked how volts are isolated. So yeah, volts are the way it works. And I actually worked in collaboration with Leonard Rome and his startup, Volt Pharma, to get bolts produced. The way that they're made is that you can actually make them in mammalian, insect, or yeast cells by expressing major bolt protein. Then, and yeast is generally the easiest. Then, you lyse the cells and you take the supernatant and you run it through a variety of different centrifugation steps, actually ultracentrifugation steps. And there's several different steps. You can look at the methods of my preprint where it describes this in more detail, but essentially you run it through a series of centrifugations and then you eventually get purified bolts at the end. Something that should be considered there is that ultracentrifugation isn't typically scalable to an industrial scale level. Although, to be fair, you can actually produce a lot more volts than you can produce AAVs on a per cost basis. So it might not be necessary to go to use a non-ultracentrifugation technique. But yeah, the ultracentrifugation. But anyway, sorry, I digress. There may be ways of developing column-based, affinity-based purification methods for purifying volts as well. It's just those haven't really been developed yet. But I think it would be relatively straightforward to do that, especially for a company. Okay, it looks like...
I also have Micah with another question. Is that okay?
Yeah, that's fine. It says the meeting ends in 5 minutes, so we'll have to wrap up. But yeah, Micah, go ahead.
Is there a way to wash out the free-floating AAV after you've done the packaging so you only deliver packages and in order to help reduce the chance of building up immunity and having immune reaction?
Yeah, that's something that I'd like to do. It's taken a backseat for now because of some of the other priorities, like getting the in vivo study done and getting the gene editing study done. But it is something I would like to do, hopefully in the near future. And the way that it would work is essentially I was able to get an anti-AAV affinity resin. And the idea is that I would then run the Volt AAV mixture through the affinity resin, and the shielded AAVs would not be bound by the antibodies on the affinity resin, and the unpackaged AAVs would be bound. And so at the end, you would only get out the bolts that have packaged the AAVs successfully, because all the other AAVs would get stuck in the affinity resin. And I actually do have that affinity resin on hand. It's just I haven't used it yet.
Awesome. Oh, how much are you trying to raise? There's another question that Micah wants the answer to.
Yes. How much am I trying to raise? Right now, I'm trying to get the animal data first, but once that has been established, I think that I'm looking to raise around $200,000 to $400,000 to get started with the preclinical toxicology and efficacy studies. And then from there, of course, there will be larger rounds to get into clinical trials. But yeah, that's where I'm at currently.
All right, then you get my final questions, which I guess Micah has already taken one, which is usually if people are excited about your project, how can they help you? If you're asking for something, like, is it collaborative? Is it that you're hiring? Is it that you're fundraising? If so, how can people contact you best?
Really, all
like
of those
useful to know.
Really all of those things. I would say there's, I'm looking for connections both to potential fundraising opportunities and to potential collaboration opportunities. I'm really open to a lot of different things. I would love to find ways of speeding up the process of getting this spun off so that it really can become something that is actually going into clinical trials and eventually becoming hopefully a approved therapeutic. And anything, either collaborators or fundraising opportunities would be great. Is there another question?
Fantastic. Micah just said maybe Lifespan Investor Network.
Cool.
Possibly too early, but worth a shot.
Yep. I'll write that down. Thank you, Micah.
Yeah. Well,
all right. Feel free to reach out to me afterwards as well.
Yeah. Logan, if you have a deck, please send it my way. Then we can try to.
Sure. Yeah.
If there's interest from some people, I at least know a few that should be interested in this stuff. Thanks a lot. Yeah. This was really interesting. That you definitely taught me a lot of new things that I had no idea existed and how they interact with each other. That was really interesting. Thanks a lot. And you're definitely doing like pretty frontier work here. So thanks a lot for that. Thanks everyone for your great questions. I really enjoyed this one. And have fun on the moon there with your 2 moons in the back, Logan.